Okay, hello Bio. Continuing on with Chapter 12, we're on Section 4 today, and this section is entitled Mutations. So what mutations are? These are things that are going to go wrong in our genetic material. If we mention all those processes from the previous section, transcription, translation, replication, those don't usually always go exactly how they're supposed to, so we call these mutations. So we're going to take a look at some specific mutations and see how they affect the body. So first question we need to ask ourselves, what are mutations? So the definition your book gives is simply changes in the genetic material. So I would write this down if I were you, know this for the quiz. The mutations are simply changes in the genetic material. So in our A's and our C's and our G's and our T's and our U's, when those get a little bit mixed up, those are going to be called mutations. And there are some specific kinds of mutations. The mutations that produce changes in a single gene are known as gene mutations. Those are going to be the ones that we look at today. And mutations that produce changes in the whole chromosome are going to be known as chromosomal mutations. So obviously, the one that just affects a single gene is going to be kind of less of a problem than the one that affects the entire chromosome. And so our first gene mutation is going to be one that involves a change in one or a few nucleotides, and those are simply known as point mutations because they occur at a single point in the DNA sequence. Pretty simple. So gene mutations involving a change in one or a few nucleotides, remember the A, the T, the C, or the G, are known as point mutations. And there are three types. These include substitutions, insertions, and deletions. So we'll take a look at substitutions to begin, and they usually affect no more than a single amino acid. So substitutions, if we look over here in the right, our DNA, should have a C and a G right here, and then for some reason, and our mutation, or when our substitution occurs, we get a T instead of an A, so that's going to give us one different amino acid, which again, usually is not that big of a deal. So substitution is just simply the changing of one single base, basically. It's just going to be one single base, and usually it's not going to be that big of a deal. However, the effects of insertions or deletions are going to be more dramatic than that, and because it's going to either be putting in a whole different amino acid or it's going to be taking out a whole amino acid. And the addition or deletion of a nucleotide causes a shift in the grouping of codons. And so changes like these are called frame shift mutations. So frame shift mutations are really just going to change the entire way that we look at our protein chain there. So frame shift mutations are going to be a lot more problematic than just a simple substitution. And how they can be more problematic is they will change every amino acid that follows the point of the mutation. So it makes, in our substitution, it made one mistake and it just changed one amino acid. In a frame shift mutation, it can make one mistake and it will change the entire chain of amino acid, which will give us a completely wrong protein. And again, that's what the next part says. It will alter a protein so much that it is unable to perform its normal functions. So frame shift mutations are bad because they can cause proteins that are completely non-functional, and then when the proteins are non-functional, the cell is non-functional, and it'll most likely end up dying. And so more specifically of what happens in an insertion, it's just an extra base is inserted into a base sequence. So if we look up here, our normal, we have TAC, GCA, TGG, AAT, then our mRNA comes along, and it's fine, and then it gives us our normal amino acids. So now in an insertion, instead of substituting our C for our T, a T simply gets stuck in by itself, and it pushes everything over. So now look, we have 1, 2, 3, 4 amino acids, and now we look, 3 of the 4 have changed now, and then now we just have one random letter floating off there by itself, which is going to do nothing. So that changes our protein completely, and that's bad news when we think of wanting our cell to function normally. So in a deletion, the loss of a single base is deleted and the reading frame is shifted. And so if we look here, this one's kind of a easier example to see because it actually gives us words. So we have the fat cat ate the rat. So now we're going to look at our deletion. Just the H gets taken out. And now look, when we try to read it, it makes no sense whatsoever. So this would be the same in genetic code, just substitute letters that we actually have in our genetic code, but they just gave this example in the book for you to actually look and see what happens when we try to read it. And this is the same thing when your cells try to read it. It just produces something that does not work whatsoever. So that's it for Mutations Part 1. Let me know if you have any questions.